Now, so let me start. Let me start introducing our guests. So let's begin with from University of the Philippines, Los Baños, an expert in spiders and other arthropods, Dr. Emilyn Barrion-Duco. Good morning, ma'am.
Morning.
Thank you, ma'am. From Silliman University, an expert in Lepidopterans, Dr. Jade Asterbadon. Good morning, sir.
Good morning.
An expert in terrestrial snails, Dr. Ryan De Chavez from University of the Philippines, Los Baños.
Good morning. Magandang umaga.
From MSU Naawan, I think he is the only expert in earthworms right now in the Philippines, Dr. Nonilon Aspect.
Good morning po sa lahat.
And of course, meron tayong surprise guest dahil hindi ko alam kung makaka-attend siya due to certain COVID reasons. No? Let me welcome from the National Museum of the Philippines, Mr. Perry Buenavente.
Magandang umaga. Salin pusa lang po ako ngayon, ha?
Hindi pwedeng saling pusa, sir. Everyone will share today. No? So, this caption now was initiated by me for my students, especially that most of them will be by their third year, kasi second years na sila ngayon, by their third year will be choosing their technical elective, hindi naman specialization, that will prepare them for their thesis. Now, most of them actually are going through the last year daw walang nag-ecology track, no? So in my bright mind, at para dagdagan pa yung trabahong naiisip ko, no, kahit marami na akong ginagawa sa buhay ko, gusto kong dagdagan pa. No, hindi ko rin alam kung anong naisip ko, no? To initiate this focus group discussion web series so that we could encourage and of course let our students learn, no, how do we do field observations, especially in ecological and diversity studies. So I've invited different experts from different taxa of interest, no, from plants to animals in different habitats, no. So let's start this focus group discussion, no. Umpisahan natin dun sa pang Miss Universe na question to our expert, no? So, I'll direct this question first to Ma'am Amy. So, Ma'am Amy, what inspired you to tackle the taxa that you are studying right now and do ecological and diversity studies in the taxa that you of your interest right now?
Actually, my parents are a big influence kasi growing up, it was my dad who brought me along his office. Tapos, my first exposure talaga, I thought yung children's toys ay insect pin, insect net, vials. Kasi I wasn't really raised to play with dolls. Yung kasunod ko kasi na kapatid ko was very close, yung age namin, 11 months. So that meant my mom had to take care of my younger sibling. Tapos ako, I was with the care of my dad. And my dad being an entomologist, although mom ko din is an entomologist, I was very much exposed to the field as early as my childhood. Nalaro ko na yung vials. Maliit pa lang ako. Way before I knew they were called vials. Siguro kumbaga familial indoctrination ang nangyari sa akin.
Ganun naman. So let's see po so with the other experts, Sir Ryan, how were you inspired to tackle your taxa?
Actually sa akin, I have first during wala akong ka-idea, wala akong childhood interest or whatsoever. Then when I went to college actually, so that's the time katulad ng mga estudyante mo ngayon who would be choosing what research field they would like to take. So during the thesis period, during my undergraduate years, I was assigned to a professor, si Dr. De Lara, who's working on freshwater mollusks. Yun yung specialization niya. And during that time, being a very obedient and very, very kind student, sige, parang masyado akong gullible during that time na, sige, mag-work ka na dito sa freshwater mollusks na to. So, sa takot ko na lang sa advisor ko, sige po, ganun na lang sinabi ko. So during my undergraduate years, I work on freshwater mollusks, more on ecotoxicology. Now going to the land snail, I was nangyari siya during my my MS years. So during that time, I was initially would like to continue what I worked on during my undergraduate, like toxicology pa rin. Pero at the middle of my program, my advisor told me, huwag ka na mag-freshwater kasi maglalaban tayo eventually in the future. So choose a group na hindi tayo maglalaban. So you work on land snails. So again, being a very obedient advisee that time. Sige, even though wala akong ka-idea, sige, it's a challenge. But anyway, that field grew on me. So marami akong discoveries, maraming mga challenges. But eventually, happy na rin ako kasi maraming exploration, maraming discoveries na nakita ko sa field na to. Kaya that's my short and brief story about kung bakit ako nag-work on land snails.
Dr. Aspe, what about you, sir? Very unusual, no, earthworms.
Actually, naririnig po ba ako?
Yes, sir.
Naririnig po ba yung boses? Okay. So yung sa akin po, actually, to begin with, I never thought na magpupurso ako ng career as a biologist. I wanted to be an artist, a professional artist, no? So graduate pa ako sa Philippine High School for the Arts sa Mount Makiling, Los Baños. And I wanted na magpupurso ng fine arts sa UP Diliman. So yun yung gusto kong track supposedly. But I didn't receive support, no, from my dad during that time. And umuwi ako and I took up marine biology. So marine biology pa and naging thesis ko pa on mangroves. So wala sa any thought na mag-earthworms pala ako later on at ito yung magiging career track ko. So when I was pursuing master's degree in biology, nagkataon na yung advisor ko, she met online a scientist, an earthworm scientist based in Kansas University who was looking for a partner, an apprentice to study earthworms in the Philippines. So, yun, nagka-link kami through my advisor. And then, at first, part-time, while I was pursuing master's degree, trabaho ko na rin yun as research assistant. So we traveled to different parts of the Philippines. And at first, I didn't realize na ganito pala ka-diverse, no? Parang just like any layman na isa or dalawang or tatlong species lang ng earthworms meron. But then when we went from one place to another, doon ako nag-start na maging fascinated in studying them. And nakita ko na, ang gaganda pala ng mga earthworm species, lalo na dito sa bansa natin, unexplored. Doon nagsimulang ma-develop yung fascination ko until kinareer ko na siya. Yun po.
Thank you, sir. Doon naman tayo sa youngest mga members ng ating panel of experts. Sir Perry, what about you? What inspired you to take on I think your expertise are ants, right? So bakit ants? And what inspired you to on them?
At my age, at 24 years old.
Sige sir, tuloy natin yung 24.
Actually, katulad siguro ng sabi ni Sir Ryan D. Chavez, masunuring bata rin ako. Na-brainwash ako ng teacher ko. I don't know if everybody knows Dr. Dave General. Nung college kasi ako, tambay lang ako sa school namin eh. So one weekend, inimbitahan niya ako to go to Mount Isarog. And sabi niya, tara, mangolekta tayo ng insekto. So sabi ko, sige, wala naman akong ginagawa this weekend. So sinamahan ko siya doon. So as the saying goes, the rest is history. So after that, nahook ako kasi ganda pala ng field biology, nakakapaglakwatsa ka every weekend. Tapos papakainin ka ng teacher mo. So ayun, nangolekta ako for a few weekends noon. And then nung inimbitahan niya ako sa lab, o sige, ito na yung kinolekta natin sa field, i-process mo. Okay, sige, pre-process ko siya. Nakakatuwa palang tingnan na ang dami nating langgam. So, sobrang dami eh. Nahook ako doon. So, from there, nagsimula na yung interest ko sa kanya. So, the same thing siguro with Ma'am Amy. Although hindi family sa akin si Sir Dave, although I already consider him as a family, medyo familial eh. Familial yung usually yung mga interest natin na nakukuha natin from yung mga nagbibigay sa atin ng inspiration. So I was inspired and then yun nga, nagpatuloy-tuloy na ako.
And of course,
pero lakwatsa talaga. Ito talaga yung lakwatsa.
Bata ba sa'yo si Dr. ba doon? Mas bata si Sir
mas matanda yan eh. Mas matanda yan sa akin. Di ba Ja, 25 years old ka na, 24 ako. I don't know.
So what about you, sir? Sir Ja, what inspired you to take on butterflies? I'm not how do I say this? I'm not being sexist, no, but it's quite unusual as well, no, for butterflies, no, being a butterfly expert in the Philippines. So what inspired you?
Okay, so I think it started during my childhood years, as young as probably 5 or 6 years old. And then living in the province, we were like, I like to play outside. I like being outside. Even though my parents won't let me go outside, I still escape the house. I just want to be in the field. And then I hang out with my childhood friends. We were collecting grasshoppers and then some plants. And then that's what we do. We just play outside. And then I became interested with insects when I went into a nearby forest. And then I saw different kinds of animals and different kinds of plants. And then after that, of course, that was when I was a child. And then you go to high school, and then in college, it was the toughest time because you have to decide what track you would like to take, what course you would like to take, because that's going to be, sometimes it will be your future or not. So I remember that when I was a kid, I enjoyed being outside. I like looking at plants and animals. I'm very curious about the natural world. So without any hesitation, I went to the registrar and said, I'm gonna take BS biology. So after for 4 years, studying all these different subjects in the biology, evolution, systematics, ecology, and then exposure in the field, I was like, this is what I like to do. And then before graduating, it so happened that my future advisor in the University of Florida came here in Dumaguete, and then he said, and I approached him that he gave a talk about the biodiversity of butterflies around the world. And then after his talk, I approached him and said, I'm also interested in, working with insects. Probably I'd like to focus with butterflies. And then when I went to the graduate school and then work in the McGuire Center and then saw all the drawers on how diverse the butterflies around the world. But when I saw the ones in Southeast Asia and the Philippines, I was just amazed. Like, wow, we have this. We have the Luzon, the Mindoro, the Palawan, the Mindanao, the Sulu, the Central, and the Eastern Visayas. I was just amazed on the diversity and the biogeography of butterflies here in Southeast Asia, especially here in the Philippines. And then I became interested in that one. I curated, and then I was able to find some new subspecies. And then it's also important that during those years, during the graduate years, I met some people and I listened to their stories, and I listened to their experiences in the field, what they do, and sometimes listening to them, you can grasp on, maybe I can also do that, or maybe we can work together. Or maybe I have an idea, but I can't do it alone. So I need to, collaboration. This is what happens when you do research. It's very, very nice to collaborate with diverse researchers from around the world. So and now I'm hooked with butterflies. So right now my focus is on the taxonomy, systematics, and biogeography, and of course the ecology of butterflies here in the Philippines, because we have almost 1,000 species, and there are still a lot of unknowns, especially the biology and ecology of this species. So it's a platform that probably needs to study in the future. So probably later on, I'm going to show you an example of an insect net. This was given to me by my advisor. This was Thomas C. Emil. So this is what I carry in the field. So that's my story on how I got into butterfly research.
Thank you, Sir Ja. So now let's go to the nitty-gritty details of how do we do field observations when it comes to terrestrial invertebrates. Last episode we were tackling on terrestrial vertebrates, and somehow there are almost similar sampling methods that are being employed in terrestrial vertebrates. Now we have a group of I think of course terrestrial invertebrates are a little bit more diverse compared to terrestrial vertebrates. So yung sampling methods din po ba with terrestrial invertebrates are that diverse as well? Ma'am Amy, what are the different sampling methods that you employ in both collecting data and collecting samples for your taxa of interest?
Nako, it's a lot. It depends on the family you are collecting and even the insect order you are collecting. I'll be leaving the Lepidopterans to Jaa. But for example, even the ecosystem you are sampling. So for example, may mga props ako dito. If we're working on leaf litter, this is a portable birdies panel na improvised by our staff using jars and even the soft drink bottles. So usually you put your leaf litter here, that's for the ground dwellers, and you put a heat source here to make sure that the ground dwelling invertebrates go down the funnel and then into a container containing a killing agent, usually alcohol. But as a practice, if you cannot bring anything along in the field, lalo na kung katulad mo ko, kalad ka rin ako, you can always bring me to the field anytime. We usually have containers or just small jars. Sometimes even my hand sanitizers containing alcohol are used as collection equipment. Standard would be, I think I have an insect net here. This would be an old insect net, circular one. And this one is a D-net, which you use for aquatic ecosystem. But you can also use the standard sweep net for aquatic systems. Kasi you cannot bring all this all the time. It will be very bulky, lalo na if you don't have anyone to carry with you. But if, for example, you're sampling spiders in crevices, if they're too small, you can use what we term as a footer. Hindi ko kayo minumura. This is a collection equipment wherein you can suck the smaller spiders from crevices. Although the disadvantage of using this one for long-term use is that you get to inhale spores also of fungi. So it's very dangerous to use this on a regular basis. I think Perry also uses this for ants, but for smaller spiders, you can use that. Another thing, apart from opportunistic sampling, is that syempre in the field, it always rains. You can also use your umbrella as a collection tool. Sabi ng mga lola natin, if you open an umbrella, malas daw pag sa loob ng bahay. But in the field, this is a very lucky equipment. You flip this over, you use the end of your insect net as a beating tool, and then you beat the hell out of the plant so that you can collect any spiders or even arthropods that are hanging on the trees. So you have rain protection, but you also have a portable collection equipment. So these are just some of my staples. We also have here cups. Our gravy containers can also be used to sample ground-dwelling spiders. So you put this along the line of your soil. Usually put one first, and then you put another one, and then you put liquid soap. Preferably something that is not colored and unscented to break the water tension. So any ground-dwelling arthropod like spiders or ground-dwelling spiders crawling in the field would shoot down here, and then you have your collection of ground-dwelling arthropods. So again, it depends on the type of arthropods you are collecting, where you are collecting, the ecosystem where you want to do the collection work, and also the behavior of your spiders. For spiders, we have diurnal and nocturnal collection. So it's a 24/7 operation. Talagang when we go to the field, we don't look human like this. I think we don't really have time to dress up, to put on makeup, to brush our hair, because most of the time we do day collection and we also do night collection. Jaan knows this. For Lepidopterans, there is day and night collection also. So for spiders, there's also day and night collection. During the day, you can use your net for opportunistic sampling. Your hands. If you're not sure about the spider, do not use your hands. And then night collection, you use flashlights. So you use to flashlight, some of the spider eyes reflect at night, so you can find them at night. So there's daytime and nighttime collection. So ang daming aspects to consider when you're looking at arthropods.
Thank you, Ma'am Amy. Speaking of different characteristics or categories to consider when sampling, Sir Perry, would you like to add to that? Especially for ants? Kasi actually for the last several episodes, marami nagtatanong talaga. Though hindi ko alam kung naiintindihan ba kung terrestrial vertebrates yung topic, pero nagtatanong sila on ants. So how do we sample for ants in particular? For both diversity and ecological studies.
Okay. Kasi maraming mga estudyante rin talagang during their thesis semesters nagpupunta sa amin kasi the museum is around the university belt, no? So you have students from LaSalle, UP Manila, UST, pupunta sa amin. Palaging sinasabi namin sa kanila, mga students, makinig kayo, you always have to ask the question first. Dapat alam niyo muna kung ano yung question nyo so that will determine kung anong methodology yung i-deploy ninyo. Okay? Kasi marami silang gustong gawin na protocol pero di nila alam kung anong question yung hinahabol nila. Sir, gusto ko mag-pitfall. Ano yung question mo? Diversity nung nasa ground level? Di, sir, gusto ko rin malaman yung nasa taas eh. So, syempre, kailangan mo rin ng ibang protocol doon. For us kasi sa ants, katulad ng sabi ni Ma'am Amy, there are actually may mga tinatawag kaming night shift sa day shift eh. So pwede kang mag-nocturnal, mag-diurnal, kasi mas maganda yung data na ma-degenerate nyo kapag nakita nyo yung delineation ng mga nocturnal foraging ants from the diurnal. And also kung nasa epigeic sila, sa ground sila, or nasa canopy sila. So always frame the question na kailangan ninyong sagutin doon sa inyong research. Number 1 yon. Sa ants, kami ni Sir Dave, we do dalawang primary protocols to collect ants. Yung na-mention ni Ma'am Amy, yung pitfall, where we put yung mga tiny cups sa forest floor. Usually 100 meters yung aming transect ni Dr. General. So may 10-meter interspersion ng mga pitfall cups kasi maximum ng mga langgam is 5-meter radius from their colony, yung foraging. So that's one. And the other methodology is yung Winkler na methodology na tinatawag, where you gather yung mga leaf litter. I don't have a sample here. Nasa office yung aking Winkler set. Basically, I can show you. Can I share my screen, Eric?
You may. Sige, sir. Pwede.
Teka lang, ha. Let me just pull out the slide. Where is that? Pwede.
Yes.
Wag magalit.
Wag lang yung mga madalas mong sinesearch. Yung sample.
Wait.
Wag yung
where is that? Shoot, nawawala yung aking slide. Ano ba yan? Where is that? Okay, sorry, may technical ako. Nawawala yung slide ko. So anyway, yung winkler kasi is basically yung leaf litter ko kolektahin mo. You will sift it in a sifter. And then parang may mga bags. Later, I can pull it out pag may ibang nagdi-discuss and then show it to you. And then, i-collect nung bag na yun, nung winkler na yun, in a collecting cup, yung mga arthropods from the siftate. So, yung dalawang methodology na yun, yung winkler and then yung pitfall kasi is the most standard for ant collection. Sila yung generally replicable across all habitat na pwede nyong gamitin na mag-compare ng data nyo across many transect. So if you put a 100-meter transect on one area of the forest and then you put another 100-meter transect in another area of the forest, in site B para site A, and then you collect both pitfall and then winkler, maganda yung data na na-generate noon.
Thank you, sir. Sir, for Lepidopterans, as compared with other arthropods, they're very motile, gumagalaw. So what's the difference when it comes to sampling methods for Lepidopterans?
Okay, so for Lepidoptera, I mean, you have diverse, for insect, you have diverse methodology when we do ecological studies. So I'm going to share what I usually do. I mean, there'll be three kinds for me to document Lepidoptera. So I'll start with butterflies. The first is, of course, the usual photography. So this is the camera that I use when I go to the field. And then I use a macro lens. It's good for macro photography with small insects. But you have, again, you have the problem on, with butterflies, they fly. So my suggestion, if you want to do a photo documentation of butterflies in an area, is that first, butterflies would be very, very abundant in an open field near a forest or just a meadow where there are flowering plants. And they're active probably early to late in the morning. So that's the time when they just go flying and they just go into the flowers. So the first thing that I would do when I do butterfly photography is don't look for the butterflies and don't chase them, because of course they will fly away. So what I usually do is I'll stand at the start of my, let's say I have an imaginary transect line, probably a let's say 200 meters by 10 meters wide. So at the start, I usually just stand and observe the habitat and observe the vegetation. And then once I see a butterfly, that's the time that I'll walk slowly. It takes a lot of time and patience to do butterfly photography, because sometimes when you do your camera like that, they'll fly. So you have to approach them slowly, and then don't just approach them and then take photographs. You can take photographs from a distance. Let's say you're 5 meters away from the butterfly. Start taking photographs, and then you go closely to the butterfly slowly and slowly until you're probably just 10 inches away from it. And then you'll be able to get or capture some detailed photographs of that butterfly for identification. Now, the next thing about photography is that sometimes when you walk towards your transect, those butterflies are probably perched already there. So when you do butterfly photography, look at the tip of the leaves. That's probably just 5 meters or 10 meters from the ground. They're usually already, they're probably perched there. So you can just carefully observe the leaves and then just look for, a butterfly that you can find. So that's what I do when I do butterfly photography. And then the next is, if you want a more intensive data gathering of butterflies, which requires a lot of energy, because in this way, you have the insect net. So with with insect net, when you do this, sometimes it's uh, it depends on the kind of habitat that you have. So if you are in a meadow, it's very easy, or in a forest edge. But the collection of your data using this determines on your speed and the length of your insect net, because this is your only range. This is your only range. And for collecting butterflies using this net, you sometimes need to be very swift, especially with butterflies that are fast flyers. For the slow ones, they're very easy to just collect. So when you go into the field, this is a bio-equipped insect net, but if you can't buy one, you can use an alambre, like a metal, and then you can use your mosquito net, and then you can just attach it to your bamboo pole. That would be an easy one. So once you made your net, when you go into the field, always put your net in this situation. Like, this has to be open. Some of you will probably, their nets are like that. You will be having difficulty collecting your insect when it's like that. So you have to do it this way. Don't do it this way because it's very difficult to maneuver the net. So I want you to do sideways, because once you miss it, you can actually just swivel the net in any direction. So this is what I usually do. But based on my experience, when you do butterfly collecting, 90% chasing, probably 10% collecting. So it takes patience. I know it's very frustrating when you want to collect that butterfly, but there are limitations when you do research, but it's fun. It's fun to chase butterflies. I mean, you can also do this with other insects, beetles, flies, and using just a simple method like this. Now, if you don't want to chase butterflies, you're just tired, and say, I don't want to chase. It's time consuming, it takes a lot of effort. I don't want to get sweaty in my OOTD attire in the field. I would need to take photographs. So there are options, don't worry. So if you don't like the net, I'll introduce you to the bait trap. Now the bait trap is another way of getting a data for butterflies, but you're only limited to frugivorous butterflies for this one. So if you want to have you don't want to chase, but you just want to set up a trap and then wait for a few days and then check what butterflies you can that was attracted to or caught in your net. Okay, Perry is actually showing his net. So this is what we have. So we tie a rope here and then put it probably 10 meters above the tree. And then as you can see at the bottom, there is a gap. There is a gap. This is where the butterflies will go in. But at the middle, there's a hole so that you can put a Tupperware and then put your trap. If it's a bait trap, you can actually put let's say a fruit. You can put your fruit here and then put it back. Let's see. Okay, let's just pretend that the fruit was there, and then you hang it, and then it will attract some of the frugivorous butterflies. Probably, let's say you can leave the trap within a day or in 5 days, and you can have several of these traps to compare different sites and compare the diversity of frugivorous butterflies in different habitats. Now, for bait traps, you can actually use shrimp. You have to standardize your. You can't just use fruits, then shrimp, and then everything. You have to be consistent. If you want to use fruits, then you use fruits for every traps that you have. You want to use shrimp, you can use that. You want to use fish, you can use that. When in my experience with
alcohol.
Okay,
Do you add alcohol?
that part. In my experience in Twin Lakes, I was trying to collect some of the rare butterflies. I am trying to document how many are they. They have to be here. So I set up a bait trap. I put pineapple slices, and then I sprinkle it with my vodka. So after that, in just a minute, I was able to attract 5 Ticandra negrosensis, the endemic ones. So you can use fruit and then you can add alcohol, but again, be consistent. If you want to add vodka, it has to be vodka, everything, not tanduay, vodka, and tequila. And I think this is normal because with Justin Nuida, with Mr. Treadaway, with Mr. Humalo, they use those kinds of traps too. Tuba, I think, lambanog. Those are good attractants for Matusini butterflies. So, okay, so if you can make your own, you can actually, I have some other traps. This is a trap, an ancient trap. This is by Mr. Nuida. Mr. Nuida gave this to me. So as you can see, they use an alambre to make this circular pattern, and then mosquito nets. And then at the bottom, they can use a plywood, or this one is just a thicker plastic. And then just put a, you know, those thin, parang manipis na alambre. And then you can put your traps there. And of course, the butterflies will just go in. So those are some of the methods for butterflies that I use. Okay, can we discuss the moth later on? I think the others will.
Sige.
Okay.
Sige po.
What happened to the leftover vodka?
To the left, tama. What happened to the
What
left?
happens to
later na.
But definitely, with other invertebrates, there would be different sampling methods. For earthworms, Sir Nonilon, what would be your preferred way of collecting your samples, and of course your other different methodologies when you collect your data in the field?
Okay, so first you have to establish, ano yung objective mo? Ano yung gusto mong malaman? If like ecological, like in an area, area affected by a lot of anthropogenic activities or human activities, pwede kang mangolekta dyan. But you will expect na ang makikita mo dyan are yung mga introduced species na. And marami yan sila. So sa mga grasslands, sa mga farmlands, and yun, sa mga gilid-gilid ng kabahayan, usually, halos mga introduced species na ang nandyan. But if what you're after is the biodiversity, to ensure that what you will collect are indigenous species, you'll have to go to areas where there are less human activities, such as in the forests. So, pagdating doon sa forest, maraming methods actually dinideploy at nababasa ko, but hindi ko masyadong ginagamit. Yung sa iba, gumagamit sila ng vibration na parang kakalugin nila yung lupa so that the earthworms would be distracted. Some other methods would use formalin. Maglalagay sila ng formalin para mabulabog yung mga earthworms underneath, and then magsisilabas, and then it's time to collect. But we find that very destructive sa ibang flora and fauna. So what we usually do with my team, again, may bit-bit lang kaming pala, mga digging materials, shovels, trowels, and yung bara. And para hindi masyadong mabigat, usually marami naman yan sa mga barangay, sa mga communities. So we ask other people, yung mga local guides namin, to improvise yung mga digging materials para hindi masyadong magbibit-bit ka ng mga mabibigat na mga pala, mga digging materials. So in the field, we establish usually either 10 by 10 meters na plot or 20 by 20. So depende whether you're sampling in a primary forest or secondary forest. But if you're working doon sa grassland or sa agro ecosystem, usually maliliit lang yung mga plots namin na ini-establish. It's because sa isang hukayan mo lang nun, you'll already get a lot of earthworms. But as what I mentioned earlier, you don't expect na napaka-diverse ng earthworms na makikita mo dyan. So going back to the forest, so if you're after like abundance, frequency, and those stuff, no, measuring their population, within the 20 by 20 or 10 by 10 plots, we dig itong mga monoliths that are like 0.5 by 0.5 and then 0.3 meters depth. So parang gagawa kami maghuhukay kami ng cake. So depende sa like if it's 10 by 10, so yung mga monoliths na huhukayin namin should be equally distributed across the plot. And then if it's 20 by 20 naman, more monoliths will be dug. So after that, okay, we have to be very careful na hindi lang basta-bastang magbungkal because may mapuputol ka ng mga earthworms, no? So you have to be very careful sa sides lang yung ikakat mo, and then you have to lift this block yung and then saka mo ika-crumble yung lupa. Himay-himayin until na yun, makolect mo yung And it's really interesting, fascinating na may mga malalaki at napaka-colorful at meron din namang mga maliliit na adult. No, highly diverse in terms of their morphology. And then, so sinisegregate namin yan based on their morphospecies, no? Morphological distinction. And then we take good photos of them. And then we take samples na, for example, gusto kang mag-culture, dadalhin mo to sa lab. So you'll have to bring live samples, no? Ilalagay mo sa isang like yung mega box na may lupa. Yung lupang pinagkunan, yung lupang gagamitin mo is the same sa kung saan ka kumuha, so that the worms will be acclimatized at hindi maninibago sa new environment. So ilalagay mo doon. But if you take samples for for morphological and like molecular examination, so you will have to kill them in usually what we do 5% ethanol, and then after some time, once na fixed na sila, then we preserve them in 95% ethanol. Ang lang, once na nailagay mo na sa 95% ethanol, so pwede mo siyang gamitin for molecular analysis later on. But once you put them in ethanol, you can expect na magdi-discolor yung mga earthworms. So on the other hand, you may also use like 10% formalin, so it stays, no? The color stays para further mo ma-describe yung external morphology niya. But you cannot expect to take samples for DNA analysis when exposed to formalin. And then so yun yung once yung for population assessment, no? But within 10 by 10 or 20 by 20, hindi mo ma-maximize yung diversity ng earthworms kasi napaka-sparse ng distribution nila. So on the other hand, for biodiversity, for species richness, usually we do yung opportunistic method of sampling na hindi na necessarily within the 10 by 10 or within the 20 by 20 plots, but kung saan-saan na lang, no? So magdidig kami, or like babalik ta rin namin yung mga rotten logs, marami niyan, no? And kung saan may mga depression, kung saan yung mga nag-decompose na mga leaf litter, yun, you can expect na may mga makukuha ka dyan sa area na yan. And then, also, doon sa, may mga earthworm species din that are arboreal, no? Na doon mo makikita sa tuktok ng puno. And we still wonder, no? Paano sila nakarating doon or what drive them na umakyat doon sa itaas. So once those have been collected for morphological examination, yun, dadalhin mo siya sa lab, and then, okay, you have to have your dissecting set because earthworms cannot simply be identified by looking at its external morphology. No? Napaka shallow ng description, ng identification, no? If just basing it through external morphology. And usually, mahirapan ka if may mga lumalapit sa akin, nagpapadala ng mga photos, magpapa-identify, and hindi ganun kadali yun na si ano yan or yan si ano yan. No, you have to, so I usually request them to bring samples, no? For internal examination. So yun, ilalagay mo siya sa dissecting pan with paraffin wax, ipipin mo yung worm, ipoposition mo dorsally, so nakadapa yung earthworm, and then ipipin mo, and then you open up the dorsal portion. No? Gagawa ka ng longitudinal section, and then like a book, flip it open, and then you see the internal morphology, and you make the necessary descriptions, no? Pwede ba akong magpakita ng mag-share ako, ha?
Sige, sir.
Okay, can you see the slides I'm showing? Okay.
Yes, sir. Yes, sir.
Yan ako lang. Okay. So this is what we do, no? So yung importanteng morphological features, no, for taxonomic identification are found in the anterior part of the earthworms, no? So ayan, yung prostomium, yun yung head part niya, yung mouth part niya, and then usually the earthworms can be identified if they're adult or still juvenile, no, by the presence of the clitellum, yung ring, as you can see here, no. So if yung earthworm wala pa siyang clitellum, then perhaps it's still a juvenile or sub-adult. So ito yung makikita, ito yung basehan mo, and only adult earthworms are good for taxonomic identification. Because sa mga juveniles, there may still be organs or parts that are still not developed. No? So you can see on the picture sa right, so you open them up in a dorsal position, and what you can see are these features. No? Like only a small portion of the earthworm, like mga 15% of its body, ang importante for taxonomic identification, because the rest of the body would already be the intestine and not very useful for taxonomic identification. And one should be very skilled, no? Just like any taxonomist is skilled ka supposedly in drawing. No? So by the way, this photo shows how earthworms mate. No? Earthworms are hermaphrodites. No? So when they mate, okay, they seek for their partner, and then they have their male pores that has penis inside, and then they extend their penis and position them towards the spermathecal pores. No? May mga yung spermathecal pores, yun yung part ng earthworms that receive the sperm. So after copulation, the earthworms depart, and both are already quote-unquote impregnated. No? And then doon sa clitellum nila, mayroong male pores dyan na magre-release ng egg. And then those egg would be covered with mucus na tatanggalin nila sa katawan nila, no? So yung mucus by moving it forward, and then when it reaches the spermatical area, i-release naman yung na-deposit na sperm from the other earthworm. And then from there, fertilization takes place. So ma-fertilize yung egg, and then continued na i-release yung mucus out of its body, huhubarin niya yun until once na natanggal niya yun, it will solidify, and then those will become the cocoon. So yun. And as what I mentioned, ito yung na dapat may skill ka rin sa drawing so that you'd be able to represent how these different organs look like. So these are some of the beautiful earthworms species that we found across the Philippines and have described. And the one on the upper left is declared to be the most beautiful earthworm species in the whole world. No? So this has been named, it's called Archiferetima middletoni. And this is found only in Baler, Aurora. No? So it is blue in color with sunny side up spots. No? May white spots and yellow spots inside. And so this is how it looks like, no? On another angle. And ginawa ko pa tong cover photo because I just love it, no? How it looks like. And this is how we do it, no? Sampling. And these are giant worms we collected here. And yun, one has to be equipped with skill with in drawings. So ganito. And mga ganitong illustrations are already publishable forms. So yun lang muna yung ipapakita ko for now.
Sige lang, sir. Pinalala sa akin ni Sir Nonilon kung bakit tumigil na ako mag-taxonomy. Kasi hindi ako magaling mag-drawing. Kaya tumigil na ako mag-taxonomy, kaya nag-ecology na ako. Kasi hindi ako magaling mag-drawing. Sir Ryan, what about land snails? How do we sample for land snails? Of course, it will depend on your question. But in general, sir, ano po yung mga sampling techniques na ginagamit po sa field?
Okay, for land snails, well, the advantage of using land snails as your model organism for your biodiversity and ecological studies is number one, they won't fly away, they won't bite you, and you can also sample even when they are dead. Okay, so the best equipment for sampling land snails is basically your eye. You have to have a trained eye because even though that they're around, you need to train your eye na, snail pala yun. Because most of them are very cryptic and they blend easily in their environment. So I think one of the best sigurong dito, parang note na narinig ko sa akong mga kaibigang malacologists is you have to think like a snail for them to for you to effectively look for them. So if I am a snail, saan kaya ako nakatira o saan ako magtatago? Okay. Now, again, going back, it again, similar to other invertebrates or terrestrial invertebrates for that matter, it would really depend on your objective. Okay. So, biodiversity lang bang gagawin mo or incidental collection or what? Okay. So, for example, if you're just like to know the diversity, opportunistic collection is okay. All you have to do is go, okay, sa isang field, for example, rainforest, and just look around the trail. Because snails would leave their shells all around. Okay, so makikita mo lang sila doon. Pero it's just a matter of analysis because, for example, some of the snails are already bio-eroded. Ibig sabihin, wala na silang periostracum, yung outer covering nila. So it would mean na namatay na sila for 3 or 5 or more than years. So yung mga intact pa yung kanilang periostracum, yung outer covering, ibig sabihin, obviously, ano pa sila? Freshly killed or kapamatay pa lang sila or bago lang sila. Okay, so again, it depends. Okay, so in the Philippines, Pinaka nasasampulan namin for land snails would be rainforest and karst or yung mga limestone ecosystem because snails are very much dependent on their microhabitats. Okay, so one important note, for example, if you're doing ecological study, obviously you have to measure different environmental variables. Okay, so you have to also measure prior to collection. For example, if you're going to a rainforest or in a karst ecosystem, you have to establish first your quadrats. For land snails, we are using quadrats. Para kasi ang aming explanation doon is para silang halaman. Hindi sila masyado gumagalaw. Okay, so the best way to capture them is to establish a quadrat. Now, the size would depend. For rainforest, we usually establish 10 by 10. Pero medyo konti yung kasi laki-laki nun. Ang hirap mag-scour ng 10 by 10. If you have, for example, 5 quadrats in a rainforest, medyo isang elevation pa lang, pagod ka na. For example, you have 10 elevations more, baka hindi ka pa matapos. For karst naman, dahil medyo rich to in calcium, medyo maliliit lang yung aming mga in-establish na quadrats dito, mga 5 by 5 lang. And we have the opportunity na dito, mas padamihin pa yung aming sample size. Okay, but make sure lang na properly distance yung mga quadrats para ma-prevent yung pseudo-replication if you really want to establish independence of replicates. Now, for sampling, sabi ko nga, kung ecological talaga yung gusto mong gawin, so again, first you have to measure different environmental variables. So, can I share my slide?
Yes, sir.
Okay, sige. Okay. So these are some of the environmental variables that we are measuring prior to collection kasi nga pag dito para intact pa yung integrity ng environmental variable kasi pag for example magsasample ka na hahanapin mo agad for example sa forest litter, disturb na yung environmental variables, for example leaf litter depth or whatever. So ang ginagawa muna namin ay nagme-measure muna kami ng different environmental variables. So elevation, temperature, humidity. So these environmental variables based from literatures, based from many studies are the ones that would really affect the survival of the land snail. So talaga ito yung gusto mong tingnan. For example, if you want to study what are the habitat requirements of these land snails, you have to establish first kung ano ba talagang habitat, kung saan mo sila nakikita. So these are some of the things that we are measuring. So for example, size of the trees, or we also measure the canopy cover using tensiometer. Okay. Now, the second thing that you have to think is there are two major groups of land snails now based on size. Okay, so we have what we call the macro land snails. These are snails that would have a size greater than 5 millimeter, and we have what we call the micro land snail. So these two pictures here are two endemic land snails in the Philippines. We have Helicostyla rufogaster, which is an example. So mga siguro mga 50 to 60 millimeter in size. Compared mo to sa kabila, which is a micro land snail, which is the recently described Hypsilostoma latispira in Masungiji Reserve. So iba yung iba yung technique sa pagkolekta nitong dalawa. So for macro land snails, madali lang. Para ka lang naghahanap, nagsi-search, pulot-pulot lang. Okay, so for safety reason, within your quadrat, we usually wear our gloves. Tapos we make use of giant forceps. Okay, para in case lang merong mga snakes or mga insekto na mangangagat, at least you are well distanced from your samples. So madali lang yan. So kolekto lang. Tapos you usually look underneath the leaves kasi ito yung usually pinagtataguan nila. And we also look underneath the logs, underneath the rocks. So yun yung mga pinagtataguan ng mga to. May mga tinatawag din tayo mga arboreal land snails, lalo na yung mga Helicostyla group, na medyo makikita mo siya sa forest canopy. So in order to collect them, usually ginagawa, pinapaakyat, o kaya minsan nagde-devise kami ng panungkit para ka na nanunungkit ng mangga. So we make use of, for example, a modified conical tube, tapos fishing hook. na telescopic. So pwedeng mong sungkitin yon para makuha yung land snail, lalo na if you want them alive. Now, for micro land snail, medyo mahirap ito kasi you really have to train your eye kasi napakaliliit talaga nila. So, ginagawa namin is, for karst environment, medyo madami sila. So, nakakapit lang sila sa bato-bato. Okay? Pero they are well camouflaged that you could hardly notice them. So, talagang titigan niya na mabuti. Okay? Pero pag nakita mo sila, napakadami naman nila. Okay? So, it's very easy to collect. So we just collect them and put them in our Eppendorf tube. Then put some ethanol if you want to do molecular work or process them later in the lab. Now, another way of collecting microland snail is you collect soil and forest litter. So usually mga 6 kilogram ng forest litter, kinukolekta namin siya sa field. Tapos dinadala namin sa lab for further processing. Ang ginagawa namin ay binibilad muna namin sila. Tapos they are parang pinapa-undergo namin ng several process of sieving. So siguro dyan na lang yan. Okay, so we, tag dito, nag-ano kami ng fractional sieving, tapos we examine them under the microscope, then saka kami mag-identify. Now, another advantage of land snail, okay, lalo na pag macro, ay may mga literatures, published literatures naman tayo sa Pilipinas na madaling tingnan para ma-ID. Okay, pero for example, kung you will try to identify a possible new species, so you go deeper. Okay, so ang ginagawa mo is aside from collecting the shell, you have to examine the internal organs. Now the question is, in order to access the internal organs, you have to crack the shell, di ba? So you have to choose the shell or the tissues or the soft tissues. Okay, so usually yun yung dilemma noon, nung unang panahon kasi you have to crack the shell para ma-access mo internal organs because for land snails, one important taxonomic character that you have to look for would be the structure of the reproductive organs. And aside from that, you have to also extract the radula or the tongue, rasping tongue, because those two are the most important in describing a possible new species. So fortunately, we have a technique right now known as Nikonuki. This is parang lang pinapakuluan mo lang siya. Parang isa-shock mo lang yung mga snails, live snails. For example, you went to an island, tapos, uy, mukhang bago to. I don't want to damage the shell, or I don't want to, pero gusto kong kumuha ng organs. So what ginagawa namin is, pinapakuluan lang siya ng certain temperature depending on the size. Tapos, ang goal kasi nun is you have to relax yung tinatawag nating columellar muscle. Ito yung muscle na naka-attach doon sa shell. So pag na-shock mo siya, lumalambot na siya, then maano mo na siya, mapupull down. So you have now an intact tissue, internal organ, and you also have an intact shell. So you can use both for your species description. So yun yung mga ginagawa namin. And another advantage pala ng mga land snails, dahil napakadami nga nila, they are very good, especially for ecological studies, really, especially if you want more sample sizes para may increased statistical power ka. So you can do many statistical analyses para ma-check mo yung habitat nila. So yun yung mga ginagawa namin for land snails.
Thank you, Sir Ryan. So we have already discussed how we do collect and how we sample the different taxa, the different terrestrial invertebrates. Now let's go to the sampling site. No, though of course this depends again. Ito yung naging papaalala na sa mga estudyante natin, lalo na pag magtitis sila. The sampling methods and the site that you will choose will always depend on the research questions that you are answering. However, in general, I will ask our panel of experts. No, how do you usually choose your sampling sites? And how do you usually delimit your sampling sites? Kasi syempre, for example, for a rainforest, malaki siya. No? So, hindi mo naman pwedeng sukalin yung buong rainforest. So, how do you choose your sampling sites? And how do you delimit where to sample? No? You may give examples na lang, ma'am, sir. For example, for specific research questions that you are tackling for a specific organism that you are studying. Now, Sir Perry, so how do you usually choose your sampling sites and delimit your sampling sites?
Sa amin kasi sa langgam, napakadali eh. Siguro sa kinauupuan nyo kapag naghanap kayo, may makikita kayong langgam eh. So, yun nga, again, depende kung ano yung question, di ba? So, if katulad ng sabi nila Sir Ryan na diversity lang yung kailangan mo, actually, kahit saan ka pumunta dito sa Pilipinas, personally sa langgam pick any mountain or pick any forested area na malapit sa inyo. I'm pretty sure may bago kayong record or bagong data na ma-generate. Because although yung taxonomic foundation natin sa langgam sa Pilipinas ay medyo merong foothold, meaning may literature tayo about yung doon sa ating mga generic diversity, species diversity, hindi pa natin nasasample lahat ng islands in the Philippines. Siguro with the exception of Luzon, a few islands in Visayas and in Mindanao, especially yung Negros, Cuernos de Negros, it was extensively sampled by Chapman. Yes, Chapman, alam ni Jia yun. So, pero aside from that, pick any mountain, pick any new habitat in your area. For ants, you will generate a new data, especially doon sa species diversity nila in that particular area. Because very few pa ang napapublish about yung species diversity in a lot of areas in the Philippines. So, pero if you are, for example, if you are an undergrad, huwag niyo pahirapan yung sarili niyo. Like, pipiliin niyo Sierra Madre kasi sobrang hirap nun. I mean, like 4 days to get there, tapos mawawala pa kayo. And then there's a problem with, siyempre alam niyo naman yung problem natin with insurgencies, di ba? So you also have to take into consideration yung mga security measures natin when we're going out into the field. Kasi I'm pretty sure alam nila Ma'am Amy to, nila Sir Ryan, Sir Noni, tapos ni Adja, na in some areas, meron tayong mga friendlies. And we really have to take care of researchers going out into the field to avoid such encounter. Although in most cases, very unavoidable yan. I don't know, Jia, kung nakwento sa'yo ni Dr. Dave Loman na when we were in Mindanao, we were surrounded by NPAs. Di namin alam. Ang sabi sa amin is they were conducting a clean-up. Pero nasa taas kami ng Mount Apo. So, kinabahan na ako. So, buti na lang, may mga friends tayo from above na are very friendly. Pero some naman are very opportunistic, kumbaga, no? So they would take advantage. So depending on what sort of research you are conducting, kung thesis lang yan, huwag niyong pahirapan yung sarili niyo. Pick a very simple area. Kasi importante lang talaga, I'm parroting my friend from the museum, si Dr. Jesmos. Ang importante kapag undergrad lang kayo is makagraduate lang kayo. So yun lang yun. Basta huwag niyong pahirapan ang sarili niyo. Pag nakagraduate kayo, nag-masters kayo, sige, pahirapan nyo yung sarili nyo. Katulad si Eric, pinapahirapan niya yung sarili niya. Yan. So, ayun nga, depending on the question, pero sa langgam, any area, bigyan nyo siya ng opportunity to collect, you will generate new data.
Thank you, Sir Perry. Ma'am Amy, sa ibang needs po?
Ang tama naman si Perry, it actually depends, at ikaw din, it actually depends on your objective. Ano ba yung gusto mong trabahuhin? Kasi one of the problems we encounter is that when students go to us for advice and I ask them, what do you want to do? They normally tell me, Ma'am, I want to study the spiders of Mount ganyan. Sabi ko, can you delimit it to a smaller group? No, I want to study everything. No, you have to be able to specialize. There's nothing wrong with specializing on a particular group. Kahit nga isang species lang siya, but you have to know the ins and outs of that particular species. That's already a valid research problem. Yung nga lang kasi we go for gold lagi We want to capture everything. I think we have that Pokemon attitude that we have to collect them all. But we also have to take note that what we are sampling are other living organisms, meaning they have lives too. So wag nating sasayangin yung buhay nila just because we don't know what we are doing at kinolekta na lang natin sila all of a sudden. by directing your attention to a particular family or a particular genus or species, that will also direct your methodologies, that will fine-tune, that will simplify everything in your life. Simplify and complicate it. Kasi nga, if for example, you decide to do orb weavers, kasi ma'am, syempre, it could be a personal objective. Malabo po ang aking paningin. Then, you choose the larger ones to sample. Kasi why bother with the micro stuff if you cannot see them in the first place? So, sample the bigger ones. Assess also your personal capability. So, when you do sampling, you also have to take note, what are your energies? How are you going to do field work? Kasi we follow also similarly with spiders like butterflies, meron tayong 2 kilometers na transect minimum that we follow. As much as possible, we stick with one elevation at a time kasi we also have limitations in terms of the number of days we could spend in the field, also considering safety ng ating team. So we have to take note na at least one elevation at a time lang, one group lang, and then you do assess your sampling effort. So kunyari, if we have a 2-kilometer line transect, when we divide this into transect points or sampling points, we allot around 10 to 15 minutes of sampling work. Para kasi minsan ang ginagawa natin sa 2 kilometer, we are excited in the first 5 minutes of our time. We devote all of our energy within the 5 minutes. By the time we reach the end of the 2 kilometers, we have no energy. 3 seconds na lang. And we make erroneous conclusions in terms of diversity kasi napagod na tayo by the end of our sampling point. So dapat we have to also maintain consistency in our efforts. At saka laging may collection permit, ha?
Oo, ma'am, yan. Lagi, problema natin yan lagi ang collection permit. Sir Ryan, sa terrestrial land snails po.
Okay, so katulad din ng sinasabi ng mga ibang panels or guests mo today. So you have to prioritize yung safety and accessibility actually. So you cannot, wag mong pagpalit yung data mo sa buhay mo. Okay, so maski ano mangyari, your life is more valuable than your data. Okay, so anyway, for land snails, a consideration, sabi ko kanina, depending on yung rainforest or sa cars, ang pinaka-suggestion ko is, ano yung pinakamalapit sa inyo? Na pwede mo, lalo na ngayon, we are in a pandemic. And I don't think na lahat tayo ay meron tayong enough opportunity to travel around, lalo sa malalayo. Okay, so ang advice ko is choose a place na accessible na pwede mo nga gawing weekender field research eh. Okay, so wag mo na masyado tayong mag-ambisyon sa malalayo, sa super remote na lugar. Okay, so yun. Then always consult your thesis advisor. Okay, kasi sila yung may wisdom eh. Hindi ka naman siguro papupuntahin ng advisor mo sa napakalaing lugar just to satisfy kung anong gusto niyang research question for you. Diba? So, again, it's a collaboration eh. Hindi siya dictatorial type dapat. Na kung ano yung gusto ng advisor yung masusunod. Dapat meron kayong suggestion, meron din suggestion advice. Pero you collaborate. So, I think that's the most effective way, lalo na ngayon sa pandemic. So, you have to find creative ways in doing not necessarily field research, pwede naman siguro mga previous collection ng advisor mo na pwede nyong gawin digitally. Katulad ngayon, ang pinapagawa ko sa mga advices ko na wala dito sa Los Baños, yung mga pictures ng mga land snails na nakolekta namin previously. Okay, then they can analyze morphological. So I think that's good enough research. Ang maganda kasi, what's your question? What's your, ano yung question na maski simple, masasagot mo at masasagot mo ng ayos. So I think that's the bottom line for doing research right now.
Sir Jaa, what about you, sir? Your idea on this?
Okay, so for working with butterflies, again, it takes a lot of chasing, but chase only when you can. Because there are others who are really eager to catch that one. You don't know, you keep running and then there's probably a cliff nearby, you might fall. And then the other problem is when you do urban biodiversity, when you try to collect butterflies in cities and urbanized areas, your other concern will be the electric wires. I mean, you have to be very careful with that one. And then if you go into the mountain, as what Amy said, we have these transect lines, and you have to do one elevation at a time, because of different elevations, you will definitely have probably a different species diversity that you're going to document or collect. So what I usually do is that there are people, oh, I would like to conduct my research on butterflies here in this. They just randomly point in some of the mountains, and then only to find out that it's a slopey area, it's a ridge. I mean, that would be very, very difficult to sample. But if you would like to sample in a forest, it would be best that you will have to do a survey first, an ocular inspection on where you are going to have your transect lines. And sometimes in the forest, especially here in the Philippines, it's very difficult to use your net, because once you catch butterflies, you're probably going to catch some ferns. You're going to be blocked by the trees. So that's the problem with using nets. So probably you can just establish a point transect that you have an open space that you are going to collect the butterflies in that area. And if you're going into a thicker part of the forest, it would be best to use bait traps. But again, it depends on your question, as what they've said. If you want to compare the diversity of frugivorous butterflies, this net or bait traps would be very, very perfect. If you want to do a general collection or documentation of butterflies, this one is the best. So I think we do the Pollard method, where we establish a transect line. Again, it depends on how long, but you have to be consistent on how many transect lines you're going to compare. So probably, let's say 200 meters long and 10 meters wide. So you're going to document butterflies, but it's okay not to be able to catch all the butterflies there, because we are already aware of that one. It doesn't matter how much effort you're going to do, you're not going to collect some of them, because some of them will be at the canopy level. So those will be very, very difficult. Now, the other thing is that when you do field work, you also have to choose the perfect day. I mean, you can also compare different seasons, but you can't, when there's a typhoon season, probably that's not the best time to go into the field, because you have all the rain, you have all probably the flooding, and probably prone to landslides. So in butterflies, of course we have summer, we do sample in the summer, also during the rainy season, but not during rainy days, just after the rain. And then for me, the best is May to June, so that you can have the transition from the dry to wet season. Because during that time, you don't get rains for days. You get a rain during that day, and then the next day, if you go into the mountains, you will see a lot of diversity of butterflies. But again, it depends on your question. If you are going to compare the diversity of butterflies in different seasons, different days, weeks, and months, then you can employ this method. So in my suggestion to those, especially the younger ones who are still able to chase a lot of things, they're very active to go into the mountains. I mean, you can work on this one and try to, sometimes when you work with insect, it takes a lot of practice on how to actually wave the net. It's not, sometimes the first time you won't be able to get the insects, they will be able to escape, but after a few tries, you will be able to, to learn how these butterflies fly, where they go, and then that's how you're going to get some perfect documentation. And then if you are going to, if you don't want to collect insects, you just want to observe, take a closer photographs. These are hampers where you use it for your laundry, and then it's collapsible. You can actually put the insect up here, and then you can actually observe them in close contact, so that you can take photographs, and then detailed photographs if you can't take a picture in the wild, because of course these butterflies will be very, very active. So you can collect and put it here and then take photographs for identification purposes. And if you do want to collect butterflies, you can use envelopes and wax papers. This is what we use for preservation. And then after that, you can freeze to prevent ants from getting into your specimens. And then there will be methods online on how to spread them. And then if you want your photographs, you can actually upload it in Philippine Lepidoptera. We have a lot of people there who will definitely help you, especially me. I'm always excited whenever there are posts about butterflies and moths. And then it's very, sometimes I would say, I don't know. It's very common with invertebrates when you say, what's this? And then we will say, I don't know. So that's very common thing, but we are always curious. With butterflies, there are always ways to work. You can also work on caterpillars, and using this is also good for rearing butterflies. If you would like to just document the life history and the biology of the caterpillar until it's adult, from egg, pupa, to egg, larva, pupa, to adult. You can have that as your probably another research if you would like to do, documentation or comparison of life histories or population of, emergence population of some certain butterflies. So again, it's okay if your research is not that complicated. It's okay that, you can do what you can do, and then your data will be very, very valuable anyway. So any kind of data is very important. New distributional record, a new variation of this butterfly, and you can probably make some discoveries along the way while doing your thesis. So that's my recommendation. Don't focus too much on finishing all this thesis and everything. That will stress you out. I just want you to enjoy what you're doing into the fieldwork, because you'll never know. That's what happened to me. I was doing all the fun, chasing butterflies and then collecting, only to find out that when an expert viewed my specimens and they said, oh, that's actually new. So while doing it, I was able to publish a short aberration of this butterfly, and it was the first. So you'll never know. This is in research. You'll never know what you're gonna get. So maybe you'll do some great discoveries, especially the moth that crawls backwards. That was my first time visiting in Mindanao in 2019. And then I was in Philippine Eagle Center, and then I was with Tristan Senarillos and Stephanie Ang, and we were just walking along the trail, observing the insects, observing the plants. And then it turns out, when I was looking at this there were a lot of ants in Philippine Eagle Center. And then I saw one thing that looks like an ant, but for me, it doesn't look like an ant. So, and then I grabbed my camera, and then I took a closer look, and I was like, it really looks like an ant, but it's still not an ant. And then I took a picture and then zoom in, and I was like, oh, it's actually a moth. And it's a moth. And what's interesting about this moth, it is because it actually walks backwards in order to mimic the ant. So and then there's a complex thing about evolutionary biology and adaptation and mimicry. And then it's not, and that was actually published just a short note in a journal, because that was first ever documentation. And I didn't do that in a very intensive way of sampling. I was just walking along the park and observing. And of course, you can do it. You can go to a nearby forest, you can go to a nearby park, and then just walk and then observe all the things that you can find there. Who knows? You can make a discovery just by doing that. And that's that's a good start of being a, in the field of natural history and biology. So that's it.
Thank you, Sir Ja. Sir Noni, for your studies.
Okay, so for earthworms, we prefer to conduct sampling during the rainy season, right after raining, because you'd get frustrated if you conduct sampling during hot season. There are a lot of instances na mauwi sa wala yung effort mo, no? So very frustrating. And another thing, you bring a lot of friends with you, no, to help you collect earthworms, because there would be a lot of instances as well. No, there were lots of days in the past, no, when I was actively collecting earthworms across the Philippines. There are indeed days na wala kami makukuha, and so with that, we value, collecting earthworms. Now, once we find earthworms, para sa amin parang treasure na, kasi ganun ka limited yung makukuha namin in a day. And sometimes yun nga, napapagkamalan kami in different places na anong kukunin nyo? Treasure. So mga treasure hunters kayo, And sinasagot na rin namin sila na, yes, we are looking for treasures and we are referring to collecting earthworms. Kasi yun nga, napaka-konti lang din nila. So if it's a funded research, go for it, no? You may need a lot of people, no? Yung mga local guides to help you with collecting samples. And also, just like what Sir Ryan mentioned, no? It's very practical na you just collect in the areas where most accessible. You don't have to go somewhere, no? And to make your findings, no, your data substantial, no, the distribution of earthworms is really very mosaic, no, and it depends on the soil sample. So better if you also collect yung mga physical-chemical parameters, like and biological parameters, no? Ano yung mga associated flora or fauna in the area, no? And also with regards to soil biochemistry, okay, you can collect yung organic matter, soil moisture content, no, pH, soil temperature, elevation, and other physical chemical parameters that you could get, so that you could associate yung diversity ng mga earthworms with their habitat. No, you'd be able to explain well na ito pa yung habitat preference ng mga ganitong klaseng earthworms. They prefer pala if mataas yung pH. And so you also have to consider, no, kung saan ka magsasampling. If you wish to collect samples in a mountain, like if you're just nearby Mount Makiling, you may also consider taking into consideration yung elevation, no? Kung ano yung elevational gradient, no, ng physical chemical parameters ng soil, and as well as the biological parameters in relation to their distribution across the elevation. So, yun po.
Thank you, Sir Noni. Now, ito yung favorite question ko na tinatanong sa panel of experts ko, which is what is your best and worst field experience? And what are the difficulties that you have faced on your recent or at least sa past field works ninyo? And how do we usually troubleshoot problems in the field? Sir Ryan, your best and worst experience sa field? Sir Ryan?
Yung siguro yung yung pinaka best and worst, siguro pareho na rin yon. Same field work experience yon. Kasi sometimes your worst would be your most memorable din, di ba? So, yung ano ko, yung during my PhD fieldwork in Polillo Island, so I have to stay there for almost a month. Okay, so since Polillo, wala masyado siyang roads, so you have to travel from one place to another using boat. So para ako naging life of Pi for 1 year, ay 1 month. So medyo ano yon. So dala-dala ko lahat ng supplies ko and everything. Tapos yung isa sa memorable, well, siguro ano lang, during my fieldwork kasi nagstay kami sa isang island sa Police, I think near Patanungan Island. Tapos I'm doing collection hanggang tanghali yon. Tapos sabi nung bangkero ko, "Sir, alis na po tayo ngayon." Sabi ko, "Bakit? Pwede naman tayo magstay dito sa island na to. Dito na tayo mag-camp out kasi medyo nandito na, tapos okay naman yung place." Tapos sabi na, "Ay, sir, ano po?" Kasi nag-park sila doon sa naglagay sila ng anchor medyo malayo doon sa pinaka-shore. Tapos nagtataka ako bakit. So pumunta ako doon. So mga na yon, mga 3 o'clock in the afternoon. Tapos, "Sir, wag po tayo dito." "Bakit?" "Kasi po yung mga tao dito, may mga killer po dito." So, after nun, so sige, diretso, travel tayo sa isang remote island, wala lang tao. Tapos wala kaming pagkain. Nang huli na lang kami ng mga crabs doon, mga crablets. Tapos pinakulo, then kanin, yun lang. Okay, so those are some of the memorable. Mahirap siya kasi parang gusgusin ka na at everything, pero you really have to tapos yung mga guide pa namin mga natives, mga agta. Ang bibilis maglakad. Ako na, tapos ang hirap ang hirap ng signal. So minsan akit pa ako ng tree house na gumagalaw. Tapos akit ako ng burol. As in, ang dami. Pero ang maganda naman doon, with those sa mga remote areas na to, ang pinaka nagustuhan ko yung mga tao. Even though that most of them nasa 5th class, 6th class municipality, they're very hospitable and they're so welcoming sa mga tao. Maski hindi nila nalalaman kung anong ginagawa mo doon. Sabi, "Anong ginagawa nyo dito?" "Naghahanap po kami ng mga suso." Tatawanan lang sila, "Suso." Parang ganon. Pero when I explain them, this is the value of these animals, ganyan, okay. Tapos pinapatuloy kami sa mga bahay ng barangay captain. Tapos paminsan-minsan inoperan kami ng mga pagkain ng mga kapitbahay. Yun. Tapos sabi ko nga, after those fieldwork, siguro nag-contribute ako ng 0.001% sa local economy nila. So, one of the anecdotes is, "Sir, salamat po sa pagkuha sa amin kasi nababili po ng uniform yung anak ko. Pasukan na yon. Tapos salamat po, nabili ko ng notebook yung anak ko. May papasko pala, may pabaon ako sa anak ko." So, yun yung mga small joys, not even scientific parang just sharing of lives with this kind of people. Siguro yun ang pinaka-most memorable during this kind of fieldwork. Aside from new discoveries na ma-explore mo sa mga remote areas na to. So yun.
I have to agree with Sir Ryan. No? Sir Noni, what about you, sir? Your best and worst and difficulties in the field? So usually, paano nyo sinasagot yung mga nangyayari sa field kapag nagkakaroon ng problema?
Okay, so dito sa Mindanao, tsaka nung nag-collect kami sa Luzon, hindi gaano ka-difficult yung mga collection namin. Napaka-importante na may bit-bit ka kasi na ethanol. And sa Visayas Islands, dyan kami medyo nahihirapan kasi ang hirap itawid na magbibit-bit ka ng ethanol from one island to another, and you'd be passing through ports, airport or sa barko. So, ang nangyayari, so bumibili na lang kami ng mga alternative like isopropyl, but pag uwi namin sa lab, later on, we would find na hindi ganun kaganda yung pagka-preserve ng mga specimens namin at nasayang lang din yung expedition namin. So, those were yung mga one of our difficulties sa sampling. But talking about, I'd like to share yung mga naging adventure namin when I was still connected with the project that was based in Kansas University. When I was in Mount Malindang in Zamboanga Peninsula, where I conducted my thesis on earthworm diversity and distribution, malapit na, there was an incident na muntik na akong tagain nung isang local na nandun. And it was a really very scary experience, no? Kasi kasabayan namin nung time na yon, nung nag-sampling kami, it was in a big program called Biodiversity Research Program. At may mga nagsasampling ng mga birds and mammals. And nakita nila na grabe yung collection ng mga birds and mammals, and then right there and then doon sa mga kabahayan ng mga Subanen, yung mga indigenous people doon, tina-taxidermy yung mga samples, and naawa yung mga natives na nandun. And then during that night, no, I tried to explain to them na this is for a bigger cause, to protect and conserve biodiversity in the area. Hindi nila naiintindihan yun. Ang mas naiintindihan nila kung ano yung ikabubuhay nila sa pang-araw-araw. So yung mga ganitong usapin, hindi nila ma-appreciate yan. So muntik ba naman akong bumunot ng itak yung isang Subanen. At sabi niya sa akin na ikaw kaya yung bubunutan ko ng mata at bubuksan ko yung katawan mo katulad ng ginagawa sa mga ibon at yung mga daga dito sa lugar namin. So nakakatakot yun. Hindi sa tinatakot ko yung mga estudyante, no? But kailangan din dito sa mga ganitong lakaran, malakas yung loob mo. No? You have to be brave. No? There was also an incident in Samar na yung isang guide namin was former commander, no, retired commander of the NPA, and then we felt secured na NPA, so marami siyang connection, so we felt safe, no, ganun yung namin. But nung pagbaba namin and babayaran na namin siya at nilipat na kami sa ibang lugar, sabi niya na gusto ko pang sumama kasi familiar din ako sa area na yun. But we would like to give opportunity also to the local people in other places. But he insisted na sasama pa rin siya kasi familiar nga siya sa lugar. And dahil sa naging argument namin, na-experience ko rin na mabunutan ng itak. No? Sa harap ko mismo, pati yung anak niya, bumunot na rin ng itak at tangka akong tagain. Kasi ba't daw ako nagmamarunong, no? So mga ganong so you really have to adjust, no, on dealing with people in different places. Marunong kang makisama. I also had the experience na nung nasa Mount Busa kami in Sarangani province, na may food supply kami for a week in the mountain, but then na-cut short mga 2 araw lang kami. It's because nagkanda ubos na yung mga canned goods namin. It's because yung mga guides namin, no, pinupuslit-puslit nila yung mga pagkain, and short na kami. So napilitan kaming bumaba ng maaga, and it was frustrating kasi may plano kayo may timetable kayong sinusunod, and di mo rin maakong magalit sa kanila kasi teritoryo nila yon. So yes na lang po, baba na po tayo. You have to be very kind, and still nakangiti ka pa rin kahit alam mo sa loob mo na naiinis ka sa sitwasyon, no? And when I was in Mount Apo also, no, I brought a team with me from Metro Manila, from Cavite, from Samar, and then nung pag-akyat namin sa Barangay Barakatan, pinigil kami ng military post doon. At sabi ng commander doon na hindi kayo pwedeng tumuloy kasi may hinahabol kami na grupo. But as young as I was, that was few years ago, I was very zealous na itutuloy po namin to kasi galing pa po kami sa iba't ibang lugar. Until I argued with them, and then sabi niya finally na, okay, enter at your own risk. So we stayed in the mountain for several days, and then yun nga, may enkwentro ngang nangyari between the military and yung hinahabol nilang unknown armed group. So kaya sa balutan kami, lahat ng ang siniguro ko lang yung pitaka ko kasi babayaran ko yung mga tao ko. Gano'n, pati yung cellphone ko at iniisip ko na sana makauwi pa kami ng safe. So yun yung happy part doon na makauwi ka lang ng safe at nakakuha pa kayo ng mga samples na you think are still unknown to science. No? Ganun yung excitement namin. I also had experience in Palawan na accidentally na baril. At buti na lang doon sa tuhod ko tumama. And kasi may kasama kaming mga bird ecologists. No? So may bit-bit silang baril. No? May yung long arm, yung rifle for yung 22, no? To collect, to shoot birds. And accidentally natamaan ako sa tuhod and I was immediately brought to the hospital and I was unconscious during they transported me to the hospital. And then the doctor said na Noy, may guardian angel ka yata kasi hindi ka tinamaan doon sa vital parts mo, no? So na-experience ko ng matamaan ng baril and yun lang na in the end, looking back, parang you still treasure those memories, no? Na naranasan ko yung mga ganitong experiences and those are enriching memories na pwede mong i-share sa iba despite but yun lang, kailangan mo talaga still importante na you keep safe wherever you go. Na priority mo talaga yung safety mo and security at makauwi ka pa na buo sa pamilya mo. Yun po.
Thank you, sir. Sir Perry, what about you, sir? Maliban sa vodka moments, your best and worst experiences in the field?
Usually, yun lang talaga yung highlight ng field work ko. Yung isang kahong tanduay. Usually, mga 3 porters yung naka-assign doon. Pero,
mahina. Sir Perry? Ay, mahina yung bandwidth ni sir. Yan ang natin. Yan ang disadvantages online. Sir Perry. Nawala siya.
Tama si Sir Noni.
Nawala. Ayun, nawala nga si Sir Perry. Sir Perry, ayan, nawala. Ayan, nawala. Teka lang.
For us, mga
ayun, nawala na si Sir Perry ng tuluyan. Nandiyan pa naman siya, buhay pa naman, pero nawala lang yung connection niya. And Sir, so before we go back to Sir Perry, Sir Jaa, your experiences in the field?
Experiences, I'm still new to this, so not that much experiences yet, but I think one of the most memorable field work was in 2019. We were in the Mount Talinis range, and then we went to this mountain, and then it was interesting because some of the type specimens of butterflies here in Negros were collected there. So when I was there, I was like, wow, this is amazing. I'd like to go back to this place. And then I was with some bird experts, and then with the guide. And then we said, and the guide said, I said that, how far is Twin Lakes from here? And then the guide just pointed with his lips. Once we ascend that and go down to that mountain, that will be Twin Lakes. So that's it. Once we climb that mountain, after that will be the Twin Lakes. And then I said, oh, we can just go there. I mean, it's very close because we can see the mountains. And then we started walking. I think we started walking around 8 a.m. So once we climb the mountain, then that will be the Twin Lakes. Then our car, the car that will pick us up back to the city will be there. And then only to find out that beyond that mountain, there are other 2 mountains that we have to climb. So started climbing around 8 a.m. I think we reached Twin Lakes around 5 p.m. I only had this for my water. We didn't have food because we thought it was going to be very close. So at the middle of the field, walking or trekking, we ran out of water, and we said they were very tired and exhausted, and we're like, I guess this is it. We're so tired, and I don't know how long we're gonna keep up. Good thing at the middle of our hike, we saw some coconut trees, and we asked our guide, "Can you get us some coconut so that we can have something in our stomach?" So our guide climbed up the coconut tree and then have something to drink, and then that one actually gave us some energy to last for the rest of the afternoon. And then when we reached Twin Lake, we were like, oh, thank God, we survived. We thought it was going to be it was going to be an easy, and it won't consume the whole afternoon just walking. So again, my advice is you have to be very clear with your guide. How far is far and how near is near? Because their near is probably far, and if they said far, I don't know how far their far is. So that's what's the most interesting. And the other thing is that when we were setting up bait traps, and when we went back, it's just a day. So we're going to get it in the afternoon and check whether we collected some specimens. And as we were walking towards the trail to get the trap, we saw three or four elementary students. They were kids, and they were eating pineapples along the way. And I and my field assistant were just staring at each other and looking. It was like, they're eating pineapples. And then when we went to the bait trap and there were no bait, someone took the pineapples. So we were thinking that those kids actually checked our traps and they actually ate the pineapples. So again, wasn't of course we didn't catch any butterflies because there were no baits anymore there. So I don't have that much experiences, or not that much experience. I started working here in the Philippines, field work, like occasionally in 2016, 2012, and then I definitely started in 2019, and then last year during the pandemic, and then right now. So probably in the future, I can share some of my, best and worst memories during my field work. So that's the only thing that I can share as of now.
Thank you, sir. Sir, let's go back to Sir Perry. Sir Perry, once again.
Okay na.
Sige, sir. Sige, sir. Yan, mukhang okay na naman. Ay, sir, medyo mabagal pa rin pala yung net mo. Ay, mabagal pa rin yung internet ni Sir Perry, sir. Sir, baka may nakikigamit ng wifi mo. Baka may nakiki-wifi sa'yo, Sir Perry. Anyway, let's go to Ma'am Amy first, ma'am. Bago pa yan. Si Ma'am Amy muna. Sige po, ma'am. Best and worst, sigurado po ako sa kanilang lahat kayo ang maraming experience.
By age? Hindi. Siguro, I can't really put things as best kasi anytime we go out, parang it's like a joy. I think a commonality between all of the panel members we have this morning is that we are happiest when we are outdoors. At pinakamahirap na nga yata itong lockdown kasi we are forced to be indoors. We are forced to stay here and make observations. Sa ultimo yung garden natin, tinitira na rin natin for observations. But one of the most memorable is spending my bridal shower in the field kasi at least a week before kasal, nasa field ako. Tapos puro lalaki kasi naman talaga ang mga kasama ko, and I'm used to that. One of the difficult things of going to the field is doing your lady business with all of those men around. Saan ka magsi-CR? What do you do if you have your period? How do you take a bath? And the like. But one trick is they're really friends. Majority pa nga of my museum partners are my kumpares. Mga kasama ko na sila. That's why it's always a joy to go out in the field. kasi they're like family to me. And they're the most gentle people I am with. Kaya I'm happiest in the field. Siguro one of the worst would be having people threaten to throw away your collections kasi hindi daw na-process yung transport permit. That was the most traumatic thing. Natalo pa niya yung nagkaroon ng kumbaga crossfire between the military and insurgents a day before we arrived there. Mas nataranta pa ako dun sa concept na itatapon dahil hindi daw na-process ang aming local transport permit. So just imagine 8 hours of doing field work, doing those collections day in, day out, night and day. And then the officials were threatening to simply throw away our collections. Na malapit na kaming bumiyahe papunta sa airport. Ganun kami nataranta noon na all that effort went into waste. Buti na lang na naagapan naman. And tama Sir Ryan, it could be your best memory also kasi it was your worst memory. I think binitbit ko din yata si Ryan to one fielding activity na lumubog yung yung bangka. Tapos ko ako pa yata magiging cause ng kamatayan ng aking colleague. Medyo kami doon kasi the boat that we usually ride on, nasira siya. So we had to choose a smaller sea vehicle. Matataas yung alon papuntang Sibuyan sa Romblon. So yung palang nakasalubong namin na isa pang boat, lumubog and all the people there died. So akala tuloy nung mga tao doon sa lugar, kami yung lumubog kasi pinuntahan na kami. At nasa balita na daw na may lumubog. So nataranta sila, kahit ako nataranta, it was the only time that I had to think about the lives of the people I am with when we are going to the field. And also because I'm a mom, mag-aalala ka na din na, naku, pwede pa lang mawalan talaga ng nanay yung mga anak ko sa pinagagawa ko. I know my family understands what I do. They respect that I'm happiest when I am out of the home, but they appreciate it very much when I am at home. So yun yung mga experiences namin. Muntik ko nang mapatay to si Ryan, kabibit-bit ko sa kanya.
Okay. Sir Perry, let's try again. Okay na ba yung
connection ka?
Sir, baka naman try sa'yo.
Okay. Anyway, regarding yung question, ano nga yung question mo?
Your best and worst, sir.
Shot?
Field. Sir, hindi shot. Wala pang vodka. Later pa. Later.
Okay. Anyway, yung best and worst, actually, kung narinig niyo yung sinabi nila nila Sir Noni, Sir Ryan, and Ma'am Amy, I mean, pag nag-field work kasi, yung mga people like us, students, listen people like us, when we do field work, we do planning before we go to the field. That is because kailangan naming plansyahin lahat ng kinks na pwedeng mangyari. Pero you always have to consider that being in the field, by nature, very dangerous talaga yon. So by the very nature of going out into the field, you are coming out of the comfort zone of yung mga traditional workplace like yung office. Kasi once in the field, kapag nag-study ka ng langgam at hindi ka marunong sa herbs, baka merong ahas na makamandag na biglang tatayo sa katabi mo. Yung pala king cobra. So second thing is maraming mga accidents that could happen when you're in the field. Usually that would entail accidents like may nahulog, nabalian, yung sinasabi ni Sir Noni na may nabarel kasi baka nakalabit yung gatilyo. Or yun nga, may mga animals talaga na by nature di natin nakikita. May kwentong nagsabi sa akin na may 2 beses daw natuklaw ng pit viper sa kanilang makeshift latrine because yung pangalawa, yung una nakalimutan sabihin na nandun sa makeshift latrine yung pit viper. Kaya yung pangalawa pumasok, natuklaw din. So, in effect, very dangerous talaga when you're going out.
Nawala si Sir Perry.
Area, yung ating natural habitat. Hello?
Nakamute ka, Eric.
Sorry, sir. Sir Perry.
Nawala ba ako?
Yan, sir. Naririnig ka na uli namin.
Yes, nawala ako.
Yan, sir, nawala ka.
Anyway, pero Hello?
Yan,
Hello, Eric?
sige sir.
Okay. Take home na lang. Take home. Yung take home message is that it's very dangerous for us to go out into the field, pero we still do it because it needs to be done. We need to collect data. We are losing a lot of our forests, and at a rate na talagang hindi na natin na replenish yung ating mga forested area, and then we're losing a lot of species. So it's very important for us to go out into the field and collect this data.
Nawala na si Sir Perry completely, I think. But anyway, so let's go to the question and answer portion of this focus group discussion. So coming from our students and our audience, no? This question is from Ana Binayog for Dr. Aspe. Ay, sorry, that's mali pala. Kay John Dale Banogon. As this is for Dr. Aspe. So, mali pala yung nababasa ko lahat. Ano ba yan? Sorry. So, John Paul Solera. So, how do you know po kung asan yung mas maraming earthworms, sir, when you do sampling?
Earthworms naturally prefer moist soils rather than dry soils. So, doon ka if you're in a forest, you look for places na mayroong depression where leaf litter are aggregating, and malaki ang chance na makakahanap ka doon. And if dry naman yung soil na pagsasamplingan mo still in a forest, in such case, you have to really dig deeper, because earthworms during dry season would just go underneath, no? Where soil is much moist. And yung earthworms din pala, meron silang yung habit, no? Yung living habit nila, merong yung anexic, epigeic, and endogeic, no? So may mga earthworms that just live on the surface, no? Feeding on the leaf litter. Meron din namang mga earthworms na vertical lang yung movement nila, and so they dig under, no? And usually doon sila nakatira underneath, no? Doon sa malalim na portion. Initially, you can distinguish them based on their color pattern, or rather their pigmentation. Usually, yung mga epigeic, yung mga nasa surface, are darker in color, whereas those earthworms na most of their time live underneath the soil, usually they are unpigmented or white. And then there are earthworms also that are anexic, that just cross across the elevation, the what do you call this? The depth of the soil, no? Umaakyat sila doon sa surface, and then bumababa naman sila underneath. So, but yeah, going back to that question, we prefer to look for earthworms in moist soils where there are a lot of leaf litter.
Thank you, sir. This question is for Dr. Ma'am Amy. No? Ma'am, what kind of permit do we need to secure when collecting insects, and how tedious it is to apply for these permits? Favorite question ata mga tao.
Actually, it's not just for insects, any wildlife, including plants. You have to secure a gratuitous permit from the DENR. But it's really very tedious. But if you already have very important that you have your prepared research proposal, kasi you also have to attach your research proposal there, because you need to clarify what are the methods you're going to be using, what sort of sampling, which area are you planning to do sampling on, kasama yung lahat sa permitting process. For undergrads, usually, naka-tie up kayo with the projects of your advisors. Kaya it's also very important. And when you are doing undergrad work, you work with a mentor who is experts in the field, kasi they do represent and they do know how to go about this permitting process. Kasi if you're studying spiders and then your advisor is not really an expert, he has no way of communicating the methodologies that are involved in submission for this permitting process. Kaya it becomes more tedious. And on another note, lalong tumatagal if you plan to do your collection work in a protected area. Kasi that means you have to attend a family meeting wherein there are members of the board you have to present your actual research to. So saka ka palang napapayagan if your methodologies are approved. And usually, they only meet on a quarterly basis. So aabangan mo pa kung kailan siya mag-meet. But then again, your advisor can also do that for you. Kaya mahalaga, tulad ng sabi ni Sir Ryan, na you are good with your mentor, with your advisor. Kasi sila naman talaga ang best friend nyo in this whole research activity. Kaya huwag nyo aawayin yung mga advisors nyo.
Mahirap naman mamawain yung mga advisor. Siguradong hindi ka makatatapos kapag
graduate.
So this question is for Dr. De Chavez. Sir, can we utilize land snails for ecotoxicological research?
Yes, pero select land snail species, since sabi ko nga kanina, most of the land snails would thrive in pristine environmental conditions. Okay, so for example, if you're trying to work on ecotoxicology wherein you have many chemicals, many pollutants, medyo more or less wala talaga sila. Pero I can suggest that you can use Lysacatina fundica, that's the common Japanese garden snail. You can use them since ubiquitous naman sila all around, especially in areas na medyo disturbed, for example, near factories and everything. So actually, for ecotoxicological studies, well, in the Philippines for that matter, mas ginagamit yung mga freshwater or marine mollusks actually over the land mollusks. So yun.
Thank you, sir. Question for Sir Jaa. Because you mentioned taking pictures in the field. So, from John Raymond Torres of DMMSU from La Union. So, when it comes to field biology observation and documentation, what is the usual pixel or clarity needed for most journal publications?
Okay, thank you for that question. Yeah, it's very important that when you submit your photographs, they have to be of high quality because they have to print it. And if you visit the journal that you are going to submit, they actually have the required, like when you submit images, there are required pixels, and they're usually higher because in publication, they want to choose a, this is why when you go to the field, you don't have to take just one picture. Take a lot of picture as much as possible. Because because those journals will actually check the quality of your pictures before they print it or before they publish it. So for me, it would, because I submitted pictures before that are of low quality, and you can actually check that. Try zooming in your pictures. If it gets too pixelated in the first two zoom-ins that you do, if it gets pixelated, that means it's probably not enough. So if you can check your pictures, you can actually check it by doing a right click and then info of the picture, and it will actually give you the information of the pixels. So based on my experiences with journal publications with photos, they really want high quality photos just for the purpose of, because some of the journals will have to print, and print is sometimes different from online publication. Of course, online will be better, but when it gets printed, they kind of sometimes the quality will change. So that's why they always prefer a higher quality, either in JPEG or sometimes in TIFF files. So I hope that answered your question. So when you take pictures, take lots of pictures so that you can choose which one to be published, and you can have other options just in case the journal will say, do you have other pictures? So that's my answer to that question.
Thank you, Sir Ja. Oh, humingi ata kami sa ng additional pictures nun sa PJSB. But anyway, I'll add to that. Usually for journal publications, especially if they're going to print your publication, usually the standard PPI is 300 PPI. So kaya nagkakaroon ng problema is it's already a very big file. So usually we just ask for Google Drive link na kasi hindi na kaya ng email. So anyway, Sir Perry, I hope you could answer this question. no? It's about sampling ants. So is there a method of collecting ants using formic acid without really harming them? Is it possible that you do not harm the ant when using formic acid?
Okay. Can you hear me?
Yes, sir.
Actually, yung question na yun, you're extracting yung chemical compounds from the ants. Here in the Philippines, familiar of any institution doing that particular study, pero abroad meron na. I'm pretty sure meron silang na-publish na methodology for that, pero currently right now wala akong paraan para mabigay yung actual protocol for extracting the compounds.
Thank you, sir.
Pero definitely, if you look up online, I'm pretty sure, especially yung mga myrmecologists studying yung compounds ng langgam, meron yan eh.
Thank you, sir. This question is from Kate Loran from the ACB in Los Baños, Laguna. This is in general for, I think, for the panel, no? In data collection, do you have or follow a set of data quality requirements and recommendations to improve the quality, completeness, and value of the sampling, and even for data sets? Sir Noni, any idea, your idea on this when it comes to data collection?
Usually so we just follow a common protocol actually. And for like quality assurance of sampling, yun yung mga the things that we need to to consider, yung species richness and yung population and how they are related with the different physical chemical parameters. It depends, no? Kung like if you want to particularly deal with a specific species, no, na pag-aaralan mo yung habitat preference or habitat requirement niya versus yung in general yung species diversity. So there are different protocols that you can follow, and I think this also applies true with the other group of organisms.
Thank you, sir. This one is from our students from University of the Philippines, Richard Parilla. Again, this question is for all of our panel members. So, of course, we are experiencing the pandemic right now. So, what can you advise students who would be doing ecological and biodiversity study for their thesis during this time of pandemic? Sir Ryan, your idea on this?
Yes, ginagawa ko na yan ngayon. I think the best siguro advice is be realistic about the situation. Wag masyado ambisyoso. Kung bawal talaga mag-fieldwork kasi nga sabi ko nga safety first. Your health comes first over everything else. So again, coordination with your advisor. Ang ginagawa ko ngayon, yung okay lang naman maski secondary data ang gamitin. Sabi ko nga, may mga previous collections kami na pwedeng i-measure. So some of my students are doing morphometrics. Tapos yung iba naman ay mga secondary data ng ecological studies na pwedeng i-process. Siguro pwede na yung gawin. Ang importante lang ay sabi ko nga is tama yung methods mo. Tapos nasasagot mo yung question na gusto mong sagutin. So yun. So yun yung kailangan nating i-take consider. So health first during this pandemic.
Thank you, Sir Ryan. Dahil sigurado kung gutom na yung panel of experts natin.
Eric.
Yes, Sir Perry.
Pwedeng mag-add ako doon sa sinabi ni Sir Ryan.
Kasi
yun nga, nasa pandemic tayo, no? And a lot of us naman are in academic institution. Tapos some of us are in museum. So the fact na we are restricted at home, madalas hindi tayo pwedeng lumabas to conduct fieldwork. Tama yung sinabi ni Dr. Ryan de Chavez na we have to process yung mga metadata analysis. Kasi usually lahat naman ng schools natin may mga collections na yan, yung biology lab. they already have an accumulated specimen-based research na accumulate throughout the years na may mga estudyanteng nangongolekta. So we are actually encouraging most mga schools, mga SUCs to invest sa natural history collection maintenance nila so that magiging parang alternative ito for everyone to gather data, first-hand data into the field, yung mga metadata analysis. So siguro it's about time that we take care of the natural history collections that the schools are accumulating into their biolabs, yung mga na-accumulate ng mga professors throughout their research na kinoconduct nila, provide them with the proper care and maintenance so that it can be useful for future generations of students na pwedeng gamitin yung information at yung data na yun.
Thank you, Sir Perry. So to generally close the session, so hopefully there are still other questions, of course, from the students, but I think our panel of experts are more than willing to answer your inquiries. Just you need to just contact them. Huwag nyo na lang silang kontakin sa Messenger kasi baka mawindang sila sa dami ng messages nila sa FB. So rather connect them through email, no? But to close this session, no, I always close this session with your advice to our panel of experts. What is your advice to our students who are interested in pursuing careers similar to us in research and field biology? So let's start with Sir Ryan. So what is your advice to our students?
Well, my advice is siguro you have to be interested first. Sabi nga is, if you are passionate about your work, money will follow later. Parang ganon. You start there. So, hindi naman na, in our field of work, which is biodiversity research, taxonomic research, etc. What drives us here, okay, keep kung ano yung nag-pursue sa amin na ba't pa namin ginagawa to. Kasi sino ba magpapakahirap umakyat ng bundok? Sino ba magpapakahirap mag-travel sa napaka-remote na islands, diba? Sino ba yung taong mag-isip ng ganun? Well, some of us are here. So, I mean, your passion should drive you. Yun. Parang, kailangan mo talagang interesado ka. Tapos, siguro, it's okay to commit mistakes. Okay? So, especially bata pa kayo. So, magkamali. So, as many as you can para learning lessons to. Tapos, choose a mentor din. Yun yung pinaka-isa sa mga importanteng life lesson ko. If you are guided by a very good mentor, I think it will make your research path a little bit clearer, not easier. Pero yun parang nagkakaroon ka ng direction. So, it will open many doors, windows, or whatever. So, yun. Tapos, isa pa is, we have to be very thankful na we are living in the Philippines because the Philippines is one of the most biodiverse country in the world. Most of the foreigners are even going to the Philippines just to do research. Diba? Tayo, nasa backyard lang natin yan diba? And there are still many organisms, not even invertebrates, other organisms na pwedeng explore. Diba? Parang, kulang actually. There are very few workers, very few scientists in the Philippines who are doing this. So, siguro, sabi nga, hindi naman lahat pwede maging medical doctor. Hindi man lahat pwede maging lawyer, di ba? Pero at this point in time, because of the many environmental issues, challenges that we're facing, I think we need more biodiversity experts in the country. So, yun po.
Thank you, Sir Ryan. Sir Perry, your advice to our students?
Okay. Siguro, tama si Dr. Ryan. It has to be driven by passion. Pero I would like to parrot lang yung advice ng aking friend from the National Museum who was also a speaker doon sa Terrestrial Vertebrate group ninyo. Number one, sa mga students, I'm pretty sure narinig nyo na to before. Wag na wag kayong papasok sa ganitong profession dahil gusto niyo sumikat. That's a no-no. Hindi ka dapat yung passion mo na sumikat ay gagawin mo stepping stone yung profession na ito. Kasi madidisappoint lang kayo. Number 2, yung profession namin, sa totoo lang, hindi ka naman yayaman dito eh. It has to be driven by passion talaga doon sa interest mo. I mean, sakto lang, hindi ka naman hihirap, pero hindi ka rin yayaman. So, yun yung advice na palagi kong inuulit kasi medyo may wisdom na rin yung si Dr. Jesmos. So, sa kanya ko palagi nakukuha yung mga advice during fieldwork eh. Pero yun nga, pick any taxon that you like, na may interest kayo. Get in touch with the person that is in that field. Mag-communicate ka sa kanya, and then be respectful as much as possible. Maraming kabataan ngayon gusto instant instant parang spoon feeding. Kailangan mo rin talagang mag-invest ng sweat and blood kumbaga in order for you to be accomplished sa field or sa profession ng field biology. Pero yun nga we need a lot of new blood kasi sa totoo lang taxonomy especially is a dying race. Very few taxonomists in the Philippines, and yun nga drive your interest with the passion that you have. So that will help you succeed. Yeah. Yun lang.
Thank you, Sir Perry. Sir Jaa, your thoughts on this?
Okay. So my advice to students is, again, as they've mentioned before, you choose the organism that you would like to focus on studying. Do not compete. It would be best to collaborate, because this is how we do research. And then it's very difficult to do research on your own without collaboration, because you try to publish things and only to find out that it's already published somewhere. So in this field, prepare to be disappointed sometimes. And then to some of you are probably eager to make discoveries. And this is my advice to the students. Most of my discoveries, I wasn't thinking about it. I didn't go to the field to make discoveries. I went to the field, I collected data, and as much as possible, I enjoyed the company, the laughters, the experiences. We fell, and then we sliding by collecting butterflies, and then all those memories. And then it's through the process, through the journey that you are working on these specimens. You may not be able to make discoveries on that one field, probably in the next. And if you do not frustrate yourself to make an immediate discovery, or do not stress yourself to publish or something like this, I just want you to enjoy. You are young, you have a lot of free time, because once you be working, you have all this bills to pay that you need to think. I miss being a student. When I was a student, I spent most of my time in the mountains and clubbing and drinking, and then sometimes I just want to go back to being student, because I want to focus. I want to go into the field. Because you're still able to go into the mountains. Once you go older, you will have limitations. So my advice to the students is, when you'll be in the field, as what they've said, do not expect to be rich, to be famous, because here we enjoy what we do. This is passion, and we are dedicating our time, effort, and money to do field research. And once you have that passion and dedication, you will definitely enjoy what you are doing, no matter what the hardships will be, no matter what kinds of difficulties. Life, as what Bon Jovi said, life ain't a merry-go-round. Life is a roller coaster. Sometimes you'll be on top, sometimes sometimes at the bottom. So again, enjoy what you're doing. and especially in this field. I hope that's inspiring enough. Thank you.
Thank you, Sir Ja. Sir Noni, dahil bata pa rin naman tayo lahat dito.
Okay, so nabanggit na rin to earlier, no? Just like what the other panel of experts mentioned, you have to be passionate sa ginagawa mo, love what you do, put your heart in it, and definitely in due time it will pay off, no? So yun nga, you have to have the right motive, hindi yung na gusto kong sumikat, gusto kong maging popular in this particular field, kaya gagawin ko to. But rather, ano lang, just keep it simple. Have the yung pagiging observant. No? Yan yung pinaka-basic eh. Na observant ka sa paligid mo, may curiosity ka. And then just like like the other pillars of biology were, no, sina Charles Darwin, the father of evolution, Gregor Mendel, the father of genetics. They were priests, mga monks, no? And they didn't expect that later in history, they will be known as the pillars of biology, no? Ang naging tulang talaga nila is yung pagiging curious nila, pagiging passionate nila sa ginagawa nila, and then everything just followed. So, yun, just love what you do, and you'd find contentment and joy with what you do.
Thank you, Sir Noni. Ma'am Amy?
Nasabi na lahat nila, pero siguro I would like to remind everyone, kahit siguro i-stalk nyo yung mga pages ng ating mga kasamahan or mga field biologists, you would notice that for the most part, you don't see a lot of their photos. Mostly yung mga earthworms nila, pictures ng landscape, pictures ng fieldwork. Kasi it tells you that if you think that this field is meant for you to become famous, popular, or a recipient of awards, then you are in the wrong business. You shouldn't be even doing science kung ang motivation mo lang is to get yourself published, to get awards, to get yourself noticed. Don't go here. Mamamatay lang kayo. I'm telling you this because the most important job as a natural history scientist or as a field biologist is to give a voice to the taxa you represent. At para you are able to represent that, you have to be able to study them carefully, observe them. Never treat a data as simple. Walang basic sa science. Meron tayong tinatawag na basic science, but the actual work is not really basic. It's a lot of hard work, physically, emotionally, mentally. Kaya sinasabi namin to kasi nothing really worthwhile is meant to be done in an easy manner. Kapag masyadong madali, don't spend your time on it. Kasi it's not really going to promote your growth as an individual. So kailangan you weigh it. And a reality is, much of what we do now may not be appreciated within our lifetime. Kasi totoo lang, you may not see us as high citation, hindi siguro kataasan ng mga H indices namin, but it doesn't mean that we are not doing impactful work. We just acknowledge that our efforts now may be useful in the near future or well beyond our lifetime. And that should be enough as a scientist to motivate you to do the right thing. And we need more scientists to be more nationalistic to work on our biodiversity. So kung makukumbinsin namin kayo, kahit isa lang dito sa grupong na to, I think we have done our job this morning. Makakain kami ng matiwasay ngayong tanghali.
Yes, ma'am, definitely. Thank you, ma'am.
Beer.
Beer. Mamaya na yung beer, sir. Mamaya pupuntahan kita dyan. Kabilang barangay lang yan si Sir Perry. But anyway, once again, thank you to our participants and of course, most especially to our panel of experts. I hope because one of the reasons why I started this webinar series, I hope I opened the forbidden door of collaboration in the Philippines. Because I think I will have to be honest with my panel of experts right now, is that we usually collaborate more with foreign scientists than local scientists. I don't know if that's correct with the same with you or not, but we usually collaborate more with foreign scientists than local scientists. And hopefully these webinar episodes will open that forbidden door of collaboration within local universities and will help, of course, a nationalistic view of how we do research in the country. So once again, thank you to all our participants, and we will see you again tomorrow. Dahil gusto ko rin pahirapan yung sarili ko, dalawa yung sessions ko bukas. I have a 10 o'clock session for marine sampling and 3 o'clock session for freshwater. Kasi yung availability uli ng mga experts. Gusto ko talaga yung mas nahihirapan ako eh.